No. Hello, everyone. My name is Ali, and I'm part of the communications and media team at the Center for Biodiversity Genomics. The month of April is Citizen Science Month, and we at CBG want to start celebrating this event by speaking with a fantastic research partner, collecting data to be sequenced here at CBG. Today, I'm joined with Dr. Erinn Fagan-Jeffries, all the way from Adelaide, Australia. Dr. Fagan-Jeffries and her team have created an exciting citizen science project called Insect Investigators. So, Erinn, would you like to start off by telling everyone about this project?
Yeah, of course. So our project was working with 50 schools in regional areas of Australia across 3 different states. And we were wanting those schools to get involved in helping to document their local biodiversity and their local insects through running malaise traps. And we were then doing some DNA barcoding, giving them some information about the species they found, and then actually passing on some of those specimens to taxonomists to see if we could find some new species that those taxonomists could describe and help the students help name, which is kind of the really exciting bit we're up to at the moment.
Oh, really, really cool. Yeah, so along those lines, on your website, insectinvestigators.com.au, I see these three key research themes for the project. So collecting, which you kind of already described, describing, and educating. So let's begin with talking about the collection process and what these malaise traps and setting them up means at the schools.
Yeah, so for people who aren't familiar with a malaise trap, it kind of looks a little bit like a tent. And it's a passive collecting trap, so insects fly into the trap and get collected in a bottle of preserving liquid. So we actually sent out 50 of these to our schools at the start of the year in Australia. So it's kind of getting to the end of summer. It's still fairly warm, but we really had, no idea how much the schools would catch or how well they would work. The schools put up the traps by themselves. We made them a little video to show them how to do it. And then they ran them for 4 weeks. So for 4 weeks over March in Australia, they collected the bottle at the end of each week of insect soup, as we called it with them. And they sent that bottle of insect soup into us at the lab at the University of Adelaide, and that's where we then started processing them.
Very cool. Yeah, so in terms of the theme of describing, have you found anything surprising? Have you found anything really new? You were talking about naming new species. That's really exciting. So talk a little bit about that.
Yeah, and this is something that's been, I guess, a little bit tricky to communicate with the schools in that we actually think probably only about 30% of Australia's insects are described. So, it's almost certain that every school has found a new species in their trap. It's more about whether we've actually got a taxonomist working on that group in Australia at the moment, and trying to get across to the schools that these specimens will be used for, tens or hundreds of years into the future potentially, and that taxonomists will be continually describing species from them. So we found lots and lots of species, lots and lots of probably new species, but what our taxonomists are actually working on at the moment is I think we've got about 10 confirmed, 10 new species that our taxonomists are currently working with schools to put a name on. So yeah, that's really cool. But we've also just been able to put some from the DNA barcode library that already exists on Bold, put some names on species that the schools have found and let them know, this specific butterfly or this specific grasshopper is currently living at your school, which is kind of nice to be able to put a name to something they might see every day in the garden.
Yeah, definitely, definitely. So you talked a little bit about your taxonomist team, and I've seen on your website that you have this awesome operations team, which includes taxonomists, school managers, but also museums too that are committed to this project. So can you tell me a little bit more about why each of these members are really essential to a successful citizen science project?
Yeah, I think citizen science projects are a lot more complex than maybe someone who hasn't done one before realizes. And we were so we really wanted to make sure that the specimens were, not thrown out at the end of the project, that even the ones that didn't get DNA barcoded went into museums and were, important scientific resources that we weren't collecting for the sake of collecting. And so having our museum collection managers and curators involved from the start of the project meant that that could actually happen, that we were making sure we were collecting the specimens and collecting data in a way that they were then usable for their collections and able to be accessioned. We wanted to make sure that the schools didn't just, put up a mobilize trap for 4 weeks, send us the samples, and then not hear from us for 6 months while we did all the processing. But putting energy into getting information back to them, getting updates, that takes a lot of time. So I was really lucky we actually had a school manager in each of the states of Australia that we were working with who made sure we were in touch with those teachers and keeping them updated. And we had discussion boards going where the schools could post the insects they were finding as they were doing other collecting, and just making sure we kind of kept that communication up through the project. We had education people involved, so we have a Science Teachers Association here in Australia, and they're involved to make sure we had our education stuff up and running. The South Australian Museum education team was there as well, helping us develop resources. And then just people to actually help work out how we're going to go from 200 bottles of insect soup to a bunch of specimens we can send to Canada and get DNA barcoded that are going to be useful for our taxonomists. That process was tricky to work out, but I was lucky to have a really awesome team involved. Yeah, big group effort, this one.
Yeah, no, that's really excellent to hear, especially about keeping all those bycatch for further research. That's really important. Um, so you've incorporated this citizen science project specifically with schools, and you've even created like lesson plans for teachers to incorporate in their classes. Can you tell me a little bit about this experience too?
Yeah, we were like I said, we really wanted to make sure that the project was more than just a 4 weeks of collecting. So we tried to come up with things that would allow the teachers to integrate the themes of biodiversity and of insects and of taxonomy throughout the year in different ways if they had time and if they could. So we worked with the South Australian Museum education team and the Science Teachers Association to come up with these lesson plans that sort of step teachers through the basics of like, what is biodiversity? How do we collect insects? What is an insect? And then how do we identify them? And how do you describe a new species? So we tried to come up with a lot of different resources that schools could pick little bits of as they had time. We were working with receptions right through to high schoolers, so year 11, 12s in Australia, which meant that the resources we made kind of had to be as broad as possible, that lots of teachers could pick them up and modify them and use them as they wanted.
Yeah, no, that's really excellent. I looked through it a little bit, and I'm definitely going to incorporate it into some children's resources too. We do Guelph Bug Day here in Canada, so that's a really excellent resource. Thanks for that. And I just want to end with this question. So, what have you learned by managing this large, complex project? Any pitfalls you've experienced, and any advice you'd give other researchers to make a successful project like this one?
I think start small is probably my good part of advice. And that's something that we did. So before we applied for the grant that allowed us to run this with 50 schools, I trialed it with 4 schools, and just, worked out what teachers needed in terms of getting it to actually work on their schoolyard, some of the issues with the time it takes to do science. Like we know how long it takes to process
Right.
specimens and get results back, and that schools operate on such short time periods that
Mhm.
you've really got to, keep them updated and make sure they know that once they send in their insect soup, we're not going to be able to tell them if they've got a new species in a couple of weeks. That process takes months, and us as scientists are used to working on those timescales, but teachers and students aren't. So I learned a lot about communication and just trial things small before you start to go big. Would be my biggest tip for someone wanting to start a citizen science project, because you learn so much just by doing.
Yeah, no, that's excellent advice. Well, thank you very much for joining us today, Dr. Fagan-Jeffries. It's really a nice opportunity to talk about your exciting work. For our audience, if you want to dive deeper into this project, I'm going to link all the social media connections, the website. I know you have Twitter and Insta and Facebook, so you can dive deeper into that. And it's just a little bit past 7 p.m. here in Canada, so that means it's around 9:15. 9 a.m. in Australia. So wherever you're watching from, good morning or good night. And thank you for joining us today.
Cool. Thanks.